So yeah, as Mike mentioned, I'm going to talk about metagenomic sequencing and how we use that here at Cambridge Technologies. So I think most people are familiar with what metagenomic sequencing is. If not, the metagenome is essentially all the genetic material in a sample. The sample being an environmental sample. So for the sake of this discussion, it's typically a nasal or fecal swab from a pig or cattle or tissues. So that sample contains genetic material from the animal as well as environment, as well as pathogens. So technology nowadays enables us to analyze that all at once. So previously we used Sanger sequencing for analyzing DNA. On the left there, the bottom left, kind of has some features of Sanger sequencing. Sanger sequencing generates one read of about 800 bases. Diagnostic labs use this, at least on the swine side, for sequencing things like the GP5 PRRS or the hemagglutinin of influenza. And the key feature of Sanger sequencing is it's targeted. You need to know what you're looking for. So when we have a sample, we need to add a primer, which is targeted towards what we want to sequence. We then sequence that via the Sanger method. We generate a read of 800 bases, and again, it's just one single read. Metagenomic sequencing uses next generation sequencing technology, and this is different because when we run a sample, we typically generate about 1 million reads. These are a little bit shorter than Sanger sequencing, being about 300 bases in length. However, we have 1 million of them. So this technology is a little bit more expensive. We're charging $250 per sample. However, as compared to getting 800 bases, you're generating hundreds of millions of bases of sequence. And the real big benefit of this is that we can do this either targeted, looking for specific organisms, or non-targeted. And really the non-targeted approach is what we're going to talk about today, and how we use that. So the other benefit of that is we can look for quasi-species. So, as we know, most viruses are kind of a swarm of viruses. There's multiple different types in a sample. So we can look at all those different variants all at once. And the other benefit is we can, instead of looking at just a region of the viral genome, we can look at the complete genome of the pathogen of interest, as well as other pathogens in the sample. So this is just a quick overview how it works. I'm not going to get into a whole lot of nitty-gritty technical detail. But when we get a sample in, the first thing we do, starting at the top, is treat that sample with nucleases, so DNases and RNases. And the idea here is that we're degrading unprotected nucleic acids. So, viral, bacterial nucleic acids protected by the coat on the organism. So, degrading unprotected nucleic acids. And then the other key to this is we use what we call random hexamer, so random primers. So again, we're not targeting a specific organism, we're just randomly reverse transcribing RNA, followed by second strand synthesis, followed by amplification. Again, this is all random. So at the bottom there, after we run this procedure, essentially we've generated millions and millions of PCR products, amplicons, based on whatever's in the sample. Next, we prep sequencing libraries based on those amplicons, and then import all that data into the assembly software, and then we perform what's called de novo assembly. So we try to piece together all those little teeny bitty reads into longer reads called contigs, and we then analyze those contigs to see what they are. So on this slide here, I've got PRRS and mycoplasma as well as pig. So we're going to see all the different viruses in the sample. We're going to see host nucleic acid. We're going to see environmental nucleic acids, things like soybean viruses. And that's based on the random approach. If we know what we're looking for example, a sample comes in, we know it's PRRS positive, and the goal is to generate a PRRS genome, we can just directly assemble those reads to a reference PRRS genome to get the complete genome of that PRRS in the sample. So this slide here is just an example of one of the emerging viruses we were working on when I was at K-State. So this sample came in from pigs with respiratory disease, and when we did the de novo assembly blast, we found this porcine parainfluenza virus. We then downloaded a reference genome. So that's what's shown in the little yellow arrows there are the reference parainfluenza virus genome. But then we mapped all those reads back to the genome so we could assemble the parainfluenza virus in the sample. So all that kaleidoscope of rainbow colors below is individual reads on the sequencer. So for this particular sample, we generated 11 million reads. Only a small percentage of those reads mapped back to parainfluenza virus, so 52,000 or 0.45%. So it seems like a small number, but just because there's so many raw reads to begin with, we still were able to generate a complete genome with 361x coverage. So again, the power in the metagenomic sequencing is really in just the sheer amount of reads that we generate and the ability to non-specifically look for essentially all the different nucleic acids that are in a sample. So the rest of this talk, I'm going to kind of go over some examples of how we've used metagenomic sequencing in the past. First, I'm going to talk about application of metagenomic sequencing for cases with unusual clinical presentation. And this is going to be where we first identified atypical porcine pestivirus. Then followed by cases where the usual suspects aren't identified. So the story here is going to be what looked like a PCV2 outbreak where PCV2 wasn't present. So clinically, something's going on. We ended up identifying this novel circovirus PCV3. The next example is going to be looking at respiratory disease. So again, kind of a complex clinical picture with a lot of different things that play a role. So again, here looking at besides the usual things like flu and PRRS, what else is there? And then finally, we're going to talk about herd profiling and feedback exposure screening to identify other pathogens in the sample. So the first example here is going to be application of metagenomic sequencing for unknown neurological disease. So this story started back in 2015 when I was at K-State. We were contacted by a producer that had lost about 700 5 to 14-week-old pigs with main clinical feature being intention tremors. So as these pigs would start to display neurological disease, it would progress. The animal couldn't walk, couldn't drink, so mortality rapidly approached 100%. Conventional diagnostics failed to identify any pathogens. And then a few months after this initial outbreak, same farm, different group of pigs, started seeing similar clinical presentation. So this was a quick video clip that was shot by the veterinarian, just to give you a reference of what was going on clinically with these animals. So what we did was we received the brain from a number of these animals. So we prepared an homogenate, performed metagenomic sequencing on that. We were able to identify a single virus, and this was a typical porcine pestivirus. Following identification in the brain, we also set up a PCR and looked in other tissues and found that they were all positive for this virus. So APPV was a virus that we had identified shortly before this study as part of a National Pork Board funded PRRS genome sequencing project. So we were just sequencing PRRS genomes out of serum, and we had identified this divergent pestivirus. So following the initial identification, we really didn't have any idea what, if anything, this virus did. What was interesting here was finding this virus in the brains of these animals with neurological disease. So APPV was just discovered less than 2 years ago. When we found it, we initially genetically characterized it. It was very distantly related to BVD and classical swine fever, so only about 25% similar. It was, however, more close related to these bat pestiviruses in China. More recently, ISU was able to show that APPV causes congenital tremors. So when fetuses are infected in utero, they are born with congenital tremors. So Again, congenital tremors have been around for a long time. The intention tremors in older pigs, I think, is relatively rare, maybe a bit of a new presentation. But again, there's a lot of questions on this virus. A lot more research is needed. What is interesting, though, is a number of groups now have shown that if you take APPV and infect sows when they're pregnant, the fetuses are born, or excuse me, the fetuses are infected in utero and they're born persistently infected. So again, I'm going to touch on this later, the use of metagenomic sequencing for feedback. But again, a number of studies have shown that APP is fairly prevalent in the swine herd in the US. So if you think about feeding back APPV positive material, essentially we're generating persistently infected pigs. So again, what's the outcome of that? You know, it's still not real clear, but again, I think it kind of speaks to the need to do a bit more diligence in characterizing samples, for example, used for feedback. And then the final point here is on the diversity of APPV. As more and more research is coming out on this virus, we are seeing that there are a number of different genotypes. So there's a lot of diversity, and we really don't understand the role of this genetic diversity in immunity. The next story is that we use metagenomic sequencing on was the identification of this new circovirus PCV3. So this was out in North Carolina. It was a sow farm with kind of chronic poor reproductive performance, so higher than typical sow mortality, higher than typical stillbirth rates. So this particular instance, there was a number of acutely dead sows with lesions consistent with PDNS. Also, a number of these sows with PDNS-like lesions were aborting. Samples went to ISU. Microscopically, lesions were consistent with PDNS, PCV2. However, PCR as well as IHC was negative for PCV2. So again, what looked like a PCV2 associated disease with no PCV2 present. So we performed sequencing on the sow tissues as well as the aborted fetuses. We identified a novel circovirus, PCV3. So that's shown on the left. So it's kind of got this typical circovirus structure, single cap and a single replicase, 2,000 base pairs, single-strand circular DNA. But what's interesting on the right there, the dendrogram, the PCV3, you can see it's more closely related to canine circoviruses than it is to PCV1 and PCV2. So for example, PCV1 and 2, the capsid genes are about 85% identical. PCV3 is only about 36% identical to PCV1 and 2. So this is a wildly different circovirus, totally or very distantly related to PCV1 and 2. Following identification of this virus, we went back and did set up a real-time PCR and looked at tissues from these animals from this farm. The sows, had fairly modest amounts of virus present. However, the mummies, we saw very high amounts of viruses, CTs in the 17 to 21 range. We also looked at just a collection of samples that were submitted to ISU for respiratory disease testing, and we found that 12.5% of those were positive. And this was just a random sample from a number of different states. The goal there just looking to see, is this a unique virus to this North Carolina farm, or is this more widespread? And it certainly looks like the latter is the case. And what was really interesting is working with Pablo Pinheiro at ISU. We went back to the ISU archive and we pulled all the PDNS cases where they failed to find PCV2 over the past few years. Of those, 94% were positive for PCV3. And we also did IHC on those and were able to demonstrate PCV3 present in those lesions. So fairly strong case for the association with PCV3 and PDNS reproductive failure. So this last slide I always like to bring up when I talk about PCV3, the first part being the PCV2 history. I think most people are familiar with this, but PCV2 was really first identified in the mid-90s in cases of sporadic PMWS in Canada. It was really the late 90s, early 2000s when we had the severe systemic disease in Europe and Asia, followed by the US. When I started in this industry in the mid 2000s, was kind of at the peak of the PCV2 PMWS epidemic, when there was millions and millions of pigs dying. So, and it really wasn't until the effective commercial vaccines in 2005, 2006, where really we got a handle on this virus. So again, I always think, let's look back to history, what's happened in the past to help guide us what we should do in the future. So with PCV3, considering those historic precedent, now we have a new circovirus with a demonstrated role in PDNS and reproductive failure. I presented data that says this virus is fairly widespread in the U.S. Swine herd. There's been a couple papers out recently in the last couple months where they've identified PCV3 in China and associated with reproductive failure. So I think it's something that we need to pay attention to and be thinking about and be proactive about. And the last point, I really don't have much data on that, but there's been a lot of talk lately about increased sow mortality across the U.S. in general. So, I throw this out there as simply something to think about. Does PCV3 play a role in that? The next example of metagenomic sequencing is looking at respiratory disease complex. So, typically we're talking about young pigs and respiratory disease. Flu and PRRS are top on the list. So this was a study with a large integrated system, and what they were experiencing was a lot of respiratory disease in pigs about 14 to about 28 days of age. And sometimes they would find flu, sometimes PRRS. A lot of times they wouldn't find either of those viruses, yet these pigs still had respiratory disease, which was leading them to fall behind. So the question was, what's going on with these pigs? Is there another virus present? So what we did is ended up sampling pigs from 3 sow farms, again, about that 14 to 21 days of age, as well as 5 nurseries, about the first week post-placement. 25 pigs per site were sampled, and we assembled those samples into pools of 5, performed metagenomic sequencing. So there's a whole lot of viruses that we found here. There's 2 of them that I highlight, porcine astrovirus 4 and porcine cytomegalovirus. These are the 2 viruses that we found most consistently at the various sites, as well as were present at the highest titers. And basically, we infer titer based on the amount of reads that we get from a virus. Before I go on, I'll just kind of mention some of these viruses. Again, we found flu, enterovirus, a number of different astroviruses, parvovirus, circovirus, parainfluenza virus, respiratory coronavirus, saplovirus. I mean, any virus that's been associated with swine respiratory disease, I think we found it here. So, and the next slide just looks at, if we look at the total reads and look at the distribution of reads based on the viruses, you can see over half the reads are associated with porcine astrovirus 4. The next virus with the most number of reads associated with it was cytomegalovirus. So again, the cytomegalovirus was identified at high levels in about 3/4 of the sites. The astrovirus was present in approximately half the sites, but at much higher levels. This was interesting because previously, when we were doing some metagenomic studies at K-State, we ran across, we found this new genotype of porcine astrovirus 4, which we presented some statistical evidence was associated with respiratory disease. So we had published this in 2016, and then when we performed this respiratory disease complex profiling study, we also found that same virus. So certainly more work's needed on astrovirus 4, but I would say circumstantial evidence is pointing towards it playing a role in respiratory disease in young pigs. This is another study where, again, it was kind of a virus, we called it a virus profiling experiment, but it was really just to look at points of concentration for swine in North Carolina. Slaughterhouses in North Carolina are oftentimes located close to commercial nursery or finishing facilities, and a lot of times have shared transport. So the system we were working with here was asking the question, hey, what happens at the slaughterhouse when we have pigs coming from multiple different sites, different ownership coming together, you know, commingling for a day or so, and then also having, you know, trucks coming in and coming out. So is this a breeding ground for virus transmission? So what we did was sample a number of different pigs, both at primary high health slaughterhouses as well as secondary cull slaughter facilities. So again, we found a whole bunch of viruses similar to the last study I presented. A lot of these viruses you probably never heard of, things like POSA virus, IAS virus, Picoburna virus. A lot of these viruses, very little is known about them. So what role they play in clinical disease is really unknown. But again, these high health pigs, besides the things like POSA virus, Picoburna virus, Tesha virus, you know, we did find some flu, some adenovirus. But what's interesting is when we looked at the cull market, so the cull slaughterhouses, we found these same viruses. But what was surprising is we found a high incidence of Seneca Valley virus, PED, Delta coronavirus, as well as identified atypical porcine pestivvirus. So it should be noted these samples were collected by a veterinarian. These animals were all fit for slaughter. So I think the take-home message here is, you know, things like Seneca Valley, PEDV in these older animals can certainly be subclinical. And if you think about, you know, transport, taking these pigs in, leaving empty, you know, as potential routes of virus transmission. So, but again, the point here is, again, that the metagenomic sequencing is a very powerful tool to identify all these different viruses. So just to conclude my talk here, you know, again, metagenomic sequencing has been around for a few years. It's starting to become more of a mainstream method in veterinary diagnostics. We offer that here at Cambridge Technologies. It's a very pliable method depending upon what your goals are. If it's simply viral genome sequencing from an isolated virus or directly from a clinical sample, both of these are very doable. It's a wonderful technique when traditional diagnostics fail to identify an etiological agent. So whether it's unusual clinical presentation or kind of a normal clinical presentation, but we're not finding the usual suspects. You know, again, going back to the PCV3 story. It's great for profiling animals and herds. You know, so we talked about the respiratory disease and the astrovirus. But also for veterinarians that are using live exposure for things like PED, rotavirus, Delta corona, PRRS, you know, as you make those tissue homogenates, you know, I think it's a good idea to take a look at the sample by metagenomic sequencing to see what else is in there before we feed it back to the breeding herd. And again, I always come back to atypical porcine pestivvirus. You know, as now that we know about this virus, we know that these pigs that are infected in utero are persistently infected. You know, I would think it'd be a good idea to look for samples to make sure that's not present, so we're not spreading that virus through the herd. And in terms of cost, you know, $250 per sample is a little bit more expensive than traditional sequencing. However, if you think about what it replaces, namely multiple PCRs, histopath, culture VI, you know, really that $250 is really on par with all those other tests if you add them up. So my personal opinion is we need to use this method more because it's very useful. And then I guess the last point is, you know, speaking to, I guess you could call it information overload. A lot of times you find a lot of new viruses or a lot of viruses that you weren't expecting, and sometimes the use of this method raises more questions than generates answers. So a lot of times, you know, I think we need to take this data and take it a step further and take a look at further studies to look at the clinical relevance of some of these viruses. So with that, I would be happy to answer any questions.